This method can be used to detect Salmonella from food samples and identify the pathogen to the strain level through genetic fingerprinting. The main advantage of this technique is that it combines Salmonella detection and subtyping into a single workflow and substantially shortens the turnaround time from Salmonella-contaminated food sample to fingerprint of the pathogen.
To begin, aseptically place a 25-gram sample of food inside a sterile laboratory blender bag with a built-in filter. Or optionally, prepare an environmental swab by aseptically moistening a sponge with enrichment broth, dragging it across a predetermined surface, and placing it inside a blender bag. Using a laboratory blender, thoroughly mix each sample with 225 milliliters of RV enrichment broth. Then, incubate the mixture at 42 degrees Celsius for 4 to 21 hours. After this, collect a 50 milliliter subsample of enrichment broth from the filtered side of the bag. Then, centrifuge the subsample at 100 times G for 10 minutes. Carefully, transfer the supernatant to a new 50 milliliter centrifuge tube, and centrifuge at 3000 to 6000 times G for 10 minutes. Discard the supernatant and wash the pellet in 5 milliliters of BPW. Resuspend the pellet in 5 milliliters of BPW. First, thaw the sample buffer and the reaction buffer from the MDA kit on ice. Place 1 milliliter of resuspended cells in BPW.